So in today's lecture, we'll be seeing about one more effective tool as far as numericals and physics are concerned. In many chapters, we'll be seeing a common thing being asked that suppose a particular physical quantity is changed, means it is increased or decreased, made doubled 3 times. Sometimes they will say percentage changes in one physical quantity, and they will ask what is the corresponding change or percentage change in other physical quantities. So these type of questions are very common and students get stuck unnecessarily in such questions. So let's see what concept is there to find changes and percentage changes, and what if the change is very small, and what if the change is not small. So what is the difference in the two cases? First case, as I told, is if the change is very small, and second, what if the change is not small, and what to do if we are asked to find sometimes change, sometimes fractional change, sometimes percentage change. So all these things will be covered in today's lecture. So I hope that all of you would be focusing on what is being said. If you focus, then definitely later on also you will find this concept being used in almost every chapter, and then you won't be having any difficulty, right? So the concept goes as finding changes. Okay. So here when we talk about changes, so suppose we say that there are two physical quantities, one I am naming as y and one I am naming as x. So if y, a physical quantity, is dependent in some way on other physical quantity x, such that if x is changed, y also continuously changes. Listen once again, if y is dependent on a physical quantity x such that if x is changed, then y is also continuously changing. So we call y is equals to fx. That is, we say y is a function of x. So if we have two variables and they are related to each other by some mathematical operation and by some mathematical equation, so we say that y is a function of x, and that is y varies or changes continuously with x. Clear? So y depends on x or y varies continuously with x. Then a change in x, suppose we change the value of x, change in x, we will mathematically represent it as delta x. Delta x, whenever you see change, listen carefully, whenever you see the word change, what do you will understand? Delta x or change means the final value minus initial value. So suppose you are saying that a particular physical quantity, its value changed from 3 to 7. That means 3 is the initial value, 7 is the final value. So the change will be 7 minus 3, not 3 minus 7. Whenever we write change, it is always the final minus initial. Clear? So then a change in x, that is delta x, will bring obviously a change in y also, because y is directly proportional, or sorry, not directly proportional, y is dependent on x, it will depend on x in a certain way as per the equation given. So it will bring a change in y also, that is, we will write it as delta y. Change in y is delta y. So delta y means y final minus y initial. Clear? Now few things that you will remember over here, very important points or golden points. So the important points that you have to keep in mind is, first thing, when they say delta y by y, change in y by y, this will be called fractional change. This is fractional change in y. If you write delta x by x means change in x by the original or initial value. So delta y by y initial we can say, and delta x by x initial. This will be called fractional change in x. So when you divide the change by the initial value, then you call it as a fractional change. Next thing, whenever you multiply fractional change into 100, whenever you multiply fractional change by 100, so what you get is, we call it as percentage change. So whenever they ask you what is the percentage change in y, you will say delta y by y initial into 100. What is the percentage change in x? Delta x by x initial into 100. So fractional change multiplied by 100 gives you percentage change. One more thing, when we talk of delta y by delta x, it is called as rate of change. There is a difference between fractional change and rate of change. Remember this thing. When we talk of delta y by delta x, it is change in y by change in x, so it is a rate of change. And which type of rate of change it is? Since you are finding for a particular interval of x from x initial to x final, this we call as average rate of change. Okay, so this is average rate of change of y with respect to x. So these are some important points which I am writing over here. So focus on this and remember this, try to remember this. Delta y by delta x will be called as the average rate of change of y with respect to x. And the fourth thing which you have studied differentiation, there have been classes of differentiation, so there also you studied. When we find the rate of change at a particular instant or at a particular value of x, we call it as differentiation. So there is another type of rate of change which is called dy by dx, differentiation. So dy by dx, what type of change is this? This is instantaneous rate of change of y with respect to x. Instantaneous rate of change of y with respect to x. Now what is the relation between the average rate and instantaneous rate? It is simple, that what happens is that when the interval in x is very small, if the interval in x, the change in x is very small, so delta x we write it as dx and delta y by delta x becomes dy by dx. So what we say is, if delta x, the change is very small, it is approaching 0, that means delta x tends to 0, this means very small change. So if the change in x is very small, the corresponding change in y will also be very small. So in that case, what we do, delta y by delta x is written as dy by dx. This is the relation between average rate of change and instantaneous rate of change. So this is going to form the basis of today's problem solving. So kindly remember, whenever we have very small changes, in that case the average rate of change, that is delta y by delta x, can be written as dy by dx. In place of delta y you can write dy, in place of delta x you can write dx. Understood? So all the points, take a screenshot and we are going to discuss all these things through questions. So you will understand how we will find the changes. Is it clear? So what is fractional change, what is average rate of change, what is instantaneous rate of change, and when do we equate the average and instantaneous rate of changes? When we say they are almost same, when the changes are very small. Clear? Okay. So now there are two important cases. What if we talk about big changes? What if we talk about very small changes? If we have big changes, you will use the normal concept, find the initial value, find the final value, and then you can find the change. But if the changes are very small, then we can use the concept of differentiation. So I am going to use a very important result. Let's see. Important result. What is that? If suppose if y is proportional to x to the power n. Suppose y, a physical quantity, depends on another physical quantity x raised to some power n. Now if this n is obviously a fixed value or a constant, then for very small changes in x, if suppose x is changed by a very small change, then we can say delta y by y, that is fractional change in y, can be directly written as whatever the power is n multiplied by delta x by x. Very important result. I will show you the proof also. You will understand. Fractional change in y will directly become the power n multiplied by fractional change in x. And we know by multiplying by 100 we will get percentage change. So this also gives us, we will say, if they ask you percentage change in y, so you will write whatever the power was n multiplied by percentage change in x. We will take examples also on this, but very important point, fractional change and percentage change, they can be related like this. The power just needs to be multiplied. So in one line you will get answer, but this concept is applicable only for very small changes, right? So let's see the proof and then we will see numericals on this and also what to do if the changes are not small. Now see, if I am saying y is proportional to x to the power n, if y is proportional to x to the power n, so can we write y can be written as some constant multiplied by x to the power n, because I am removing the proportionality and we are using a constant c. So y can be written as c x to the power n, right? Now what we know, if I differentiate it, you all have studied differentiation, so can I write dy by dx? All you know what is the formula for dy by dx? If you have x to the power n, whenever we have x to the power n, the differentiation is what? N x to the power n minus 1. Do you all remember this? So y is c x to the power n and dy by dx is this. Clear? Now see, if I take dy on one side, I will write c n x to the power n minus 1 into dx. Is it clear? Just see, I have written it on one side. Let's call this equation as 1. Can I write like this or not? Reply fast. Okay. Now what I am doing is just divide this by first equation. Divide by first equation. So if you divide by first equation, so what you will get? Dy divided by y. Right hand side you have Cn x to the power n minus 1, and here on right side you have C x to the power n. Now what will happen here? C cancels and one more thing is missing, that was dx. Okay. Now see what we are getting. Dy by y is equals to n x to the power n minus 1 by x to the power n. Can I write it as x only? Tell me, do we get this or not? dy by y is n dx by x. Now in the just previous case, I am just taking 1 second to go back to the previous page. I said when the changes are very small, dy can be written as delta y and dx can be written as delta x. Remember, dy can be written as delta y, dx can be written as delta x. So if we come back for very small changes, dy will be delta y and dx will be delta x. So what we are getting? Delta y by y is n times delta x by x. Proved. This is what we were saying. Fractional change in y is equals to n times fractional change in x. Understood? So this relation you have to remember whenever we are dealing with very small changes, directly multiply the power. But kindly never use this thing if the changes are big. So it will be clear from the examples which we are going to use now. So write it or take a screenshot fast. Clear? Okay. One more thing, when you are not having small changes, so obviously you won't be using this thing. What you have to remember is sometimes students struggle with the questions on percentage. For him, please wait. I told what if changes are big, I will go with examples. I told if the changes are big, then you will have to normally find initial value, final value, and then you can find the change. I'll take examples. Just don't lose patience for some time. Just wait. We will come to know about it. What I am trying to say, one more thing is, whenever you see that in some questions they give percentage changes. So if suppose the changes are not small, they are big and they have given percentage changes. So what is the meaning of percent? Try to decipher the meaning of percent. Percent means per cent. Cent word is used for 100. Per 100. So basically when you have questions based on percentages and the changes are large, so what you can do is you can always take the initial value as 100. You can always take the initial value of x as 100. So accordingly you will get the final value very simply. For example, suppose I say that x, when I said y is proportional to x, x suppose initially I take it as 100. They say that x changes by 30%. What does that mean? If initially you took 100, now new value will be 130. If they say x decreased by 20%, what will you say? Initial was 100, final is 80. See how simple it is. If they will say x got increased by 200%, increased by 200%. So that means if x initially was 100, the final value will be 300, increased by 200%. So you will add 200 more. Understood? So this is what we do. Yes. So percent means per 100. So this small thing kindly try to remember. Now let's come to examples. Now first example, you all very well know about kinetic energy. What formula we have? Kinetic energy formula we know is half mv square. Half mv square. And you also know kinetic energy and momentum, very commonly asked question, so I am using this. Momentum we know the formula as mv. So if you substitute over here, so what can I say? Can I write kinetic energy is equals to P square by 2m? Kinetic energy is equals to P square by 2m. Can I write so? Kinetic energy is P square by 2m, right? Now, yes, P square by 2m. This is the relation. So can I say if mass is kept fixed, this means kinetic energy will be proportional to p square, or can I say momentum will be proportional to root ke? Can I write these two or not? If mass of the body is unchanged, it is not increased or decreased, so can I say that kinetic energy will be proportional to p square and momentum p will be proportional to root kinetic energy? Okay, so let's now go for changes. First part we are having, find percentage change in kinetic energy if momentum is increased by 20%. If momentum gets increased by 20%, what is the percentage change in kinetic energy? Now, is this a small change? You cannot say it is a small change, it is 20% a big change. Okay, so 20% is a big change. So now how will you do this? See, we are seeing kinetic energy is proportional to p square. So we can say that Ke is proportional to p square. That means, can I write final kinetic energy by initial kinetic energy is equals to p final by p initial whole square. Now, what I just said, if you take, they are saying momentum is increased by 20%. Can I take initial momentum as 100? If I take initial momentum as 100, then the final, can I write it as 120? Suppose I am taking it as 100. As I said, in percentage you can take the initial value as 100. So final will become 120 or not? Clear? Yes. Now see, this this becomes how much? 144 by 100. 144 by 100, is it clear? Now with the blink of an eye, I can say what is the change in kinetic energy. Now just see, what is the initial kinetic energy? 100. What is the final kinetic energy? 144. So how much did the kinetic energy increase? Tell fast. So kinetic energy final by kinetic energy initial, you can see 144 by 100. So if the initial value you are seeing as 100, final is 144. So what is the percentage? What is the percentage? Yes, 44. Simple. So we can say that therefore percentage increase in kinetic energy is equals to 44%. Now those who did not get it, you can understand it in the way which we have already seen. Let's see. If you did not get that how it is 44%, how can we think? So you can say also alternately, I can say Ke final by Ke initial is this. So percentage, for that we have to find delta Ke by Ke initial. Now what is delta Ke by Ke initial? Delta Ke by Ke initial means Ke final minus Ke initial by Ke initial. Now if I divide, it becomes Ke final by Ke initial minus 1. Now what is Ke final by Ke initial? It is 144 by 100 minus 1. It becomes 44 by 100. So if it is fractional change, so percentage change in kinetic energy just multiplied by 100. So after wasting a lot of time, you get the same answer which you got directly here. Is it clear? So percentage change in kinetic energy is 44%. We get like this also. Change in kinetic energy by initial kinetic energy into 100. But directly you can see that Ke final by Ke initial is 144 by 100. So from here, if initial value is 100, final value is 144, what is the change? 44. Simple, in one line you got this. Is it clear? Please let me know fast. Okay. Now, Mallikarjun is, sir, is this useful for all questions? Then I am telling something, obviously it will be for use only, then that's why we are telling so, right? So focus, you will see when you will be studying the chapters, you will see whether it is useful or not. Okay, and that's upon your wish. If you find that no, I don't want to use all these things, I can solve it in any other way, then you can go with that. Best of luck. So next we are taking again, if kinetic energy of a body decreases by 3%, find the percentage change in momentum. Now reverse. Kinetic energy of a body decreases by 3%, find the percentage change in momentum. Now let me know, is this a large change or a small change? Is this a large change or a small change? Small change. So for small changes we can go directly by the concept which I told. So what we can say, since you know for small changes what we can use, now they are giving kinetic energy and they are asking momentum. Can we say momentum is proportional to root ke? Root ke means kinetic energy to the power 1 by 2. So what will I say? Directly, without any discussion, percentage change in momentum is equals to, just multiply the power, half percentage change in kinetic energy, because it was a small change. So why we have used this? Because it is a small change. Now focus on one thing, percentage change in kinetic energy, they are saying it is decreased, it is decreased. So this will be minus 3%. So when you divide by 2, you get minus 1.5%, that is decreases by 1.5%. So the momentum in this case is going to decrease by 1.5% directly. So when we have small changes, so here we saw it was a small change. So that's why percentage change in P I have written as directly power multiplied by this. Is it clear? Okay. So understood, when we have big changes, when we have small change. Now let's try it with some other examples. You will study different chapters, they are there in physics. You will study about gases, you will study about gravitation. So we are using some portion from these chapters and we will put it in form of a question. So you are, or maybe you may not be knowing some of the laws, but when you will study that chapter, you will come to know that yes, this is actually a law. But presently I will be just adding the law so that you can know that yes, how I can find the percentage change if this law is valid. Okay. So let's see. Next question we have, for planets or for satellites, you know satellites revolve around planet, revolving around a planet. You will come to know in gravitation chapter, I am telling the name also, for satellites revolving around a planet in circular orbits. You will come to know of a formula, T square directly proportional to R cube. This T is known as time period of revolution, and R is known as the orbital radius. The orbit radius of the satellite and the time period of its revolution, they are related as T square directly proportional to R cube. So this is a relation which you will study in gravitation chapter. Now, if the satellite is shifted to a new orbit of radius 4 times the previous orbit, find percentage change in its time period. If the satellite is shifted to a new orbit of radius 4 times the previous orbit, find the percentage change in its time period. This is the question. Okay. So now, how to solve this? The change is not a small one. You are making the radius 4 times. It is a big change. So what we do, and second part also we are adding to the question. If the radius is increased by 1%, what will be corresponding increase in time period? So 2 examples we are taking. So what you see from here is, first of all I told that T square is proportional to R cube. This is our main formula. If T square is proportional to R cube, that means time period will be proportional to R to the power 3 by 2. Now if you see the first one, the change is not small. The radius has been made 4 times. So what we can say, first case, it's not a small change. T final by T initial will be R final by R initial to the power 3 by 2. Now they have said that the final radius is made 4 times the initial. So it will become how much? 4 to the power 3 by 2. 4 to the power 3 by 2 means what? 4 can be written as 2 square power 3 by 2. So this becomes 2 cube, that is 8. So T final by T initial is 8. So what is the percentage change they are asking in time period? So what we can say from here, T final will be 8 times T initial. So percentage change in time period is delta T by T initial into 100. Now what is delta T? Change. Final became 8 times. So what is the change? Change is 7 times. So how much it becomes? 700%. So final time period became 8 times the initial. So what you will say, what is the percentage change? 700%. Because it increased by 7 times. It increased to 8 times, that means it got increased by 7 times. And 7 times means 700%. Whereas the second part, it is very simple. They are saying the radius has increased only by 1%. Second case, change is very small. So if the change is very small, then percentage change in R is very small, so directly you can go percentage change in time period will be equal to just multiply the power. T was proportional to R to the power 3 by 2. So 3 by 2 times percentage R. They have given R has increased by 1%. So this will be direct 3 by 2%. That means 1.5%. So if the change is very small and the radius got increased by 1%, then the time period will increase by 1.5%. Understood? So these type of questions, when they ask you, you just need to know whether the changes are being made, they are small or they are large. If they are very, very small, then you can go directly by this concept multiplying the power. And if you see that changes are not small, then what is the concept? Final by initial. And sometimes if you see percentage, then I told you can take the initial value as 100, final value you can correspondingly write, then you can get the change. Okay. So this is all about percentage and fractional changes. I hope you got an idea of what to do. You will see this thing, believe me, in most of the chapters, finding changes, and it is a very common question, right? So that will be all. Thank you.
So today we are going to start another lecture of structural isomerism. In last class, what we have discussed about some basic functional groups, how to find total number of structural isomers for alcohol, for amines, for ether, and same primary, secondary, tertiary alcohol, how to find out. So their representations are very important. Now in this class we'll cover some more functional group, that is carboxylic acid, ester, amides, benzenoid, that is aromatic isomers. So let us start with first functional group, carboxylic acid and ester. So question is, find total number of structural isomers of carboxylic acid and ester having molecular formula C5H12O2. Because in carboxylic acid and ester, how many oxygen are present? 2 oxygen atoms are present. So for C5H12O2, what we have to find? Carboxylic acid and ester. Now easier is carboxylic acid. In ester we have to take different cases. So representation is important for ester as well as carboxylic acid. So let us first start with carboxylic acid. Now what is carboxylic acid functional group? COOH. So what is representation? RCOOH. So which is fixed part here? COH is fixed part for carboxylic acid. Only what is variable part? That is alkyl. Now total 5 carbon atoms are present in this molecular formula. So one is already involved in COH. So how many are remaining? 4. And how many hydrogen are remaining? I think I have written C5H10. I have written C5H12 because 2 oxygen are also present. Now one is unsaturation. So C4, how many hydrogen are present? 9 hydrogen. And how many oxygen are present? 2. So C4H9 has how many isomers possible? 4 structural isomers are possible for C4H9. Clear? So if they ask how many only carboxylic acid are present, so carboxylic acid are only 4 isomer. Now in case of C4H9 butyl, there are 4 isomers or alkyl present. Normal alkyl, secondary alkyl, tertiary normal, and next is secondary butyl, tertiary butyl, and one is isobutyl. So 4 isomers of C4H9 butyl is possible. Now next is for ester. Ester is important. Now representation for ester is C double bond O, OR, and on this side either R can be present or hydrogen can also be present. So this is very important. With C double bond O on this side, either alkyl group can be possible or hydrogen is also possible. And on this side, what is possible with oxygen? Always alkyl. We cannot write hydrogen, because if you write here hydrogen, it will be converted to carboxylic acid. That is not ester functional group. So if we have to write for C5, so let us first start with hydrogen on this side, because smallest can be hydrogen. C double bond O, oxygen. Now one carbon is involved, so how many carbon are remaining? 4. C4H9. And for C4H9, how many isomers are possible? 4 structural isomers are possible. Now next case we can take on this side, what we write CH3. So let us write here CH3, C double bond O. Now how many carbon are involved? 2 carbon, and total are 5. So remaining are how many carbon? 3 carbon, C3H7. And for C3H7, how many isomers are possible? 2 isomers are possible for C3H7. Now next case can be, in place of CH3, what we can take? C2H5. So next case I am writing C2H5, C double bond O, oxygen. Total 2 plus 1, 3 carbon are involved. So remaining are how many carbon atom? 2. Now in C2H5 there is no isomerism. So this is only one structure. This is only one structure. Now next is very important. We can take here C3H7, C double bond O, OCH3. Now listen, these two are not identical, these two are different. Here oxygen is bonded with propyl group C3H7. Here oxygen is bonded with CH3. So that's why they are not identical, they are different. In case of ether, what we are considering, both are same, because oxygen, both side alkyl are bonded with oxygen. But here one alkyl group is bonded with C double bond O and one alkyl is bonded with oxygen. So that's why these two are different. Now if we write, how many isomers are possible for C3H7? 2 structural isomers are possible. Now again, if you write here C4H9, then there it will be hydrogen. That is not possible, because hydrogen, if you write here, it will become carboxylic acid. That is not ester. So total how many ester are possible? 4 plus 2, 6, 6 plus 2, 8, 8 plus 1, 9. So total number of ester are 9. And total number of carboxylic acid are 4. So in question, if they ask how many total isomers are possible, so what is answer? 4 plus 9. How much it is? 13. So this is answer for this question. Total number of carboxylic acid and ester possible for formula C5H10O2. 4 are carboxylic acid, 9 are ester. So total it is 13. Any doubt in this? Check this question. Here, let us move to next part. Now this is functional group carboxylic acid and ester. Now next functional group we will discuss about aldehyde and ketone. So let us move to next. Question is, find total number of structural isomers of aldehyde and ketone having molecular formula C5H10O. Okay. Now because in aldehyde and ketone only one oxygen atom is present. So this we have to solve. So again what we write? First for aldehydes, then we write for ketones. So let us first start with aldehyde. Now we all know that which functional group is aldehyde? CHO. If this is present, CHO, this functional group is called as aldehyde. So if we write C5H10, total how many? 5. One CHO is fixed in aldehyde, means one carbon. So remaining how many carbon are present? 4. And how many nitrogen are there? 9. So C4H9. And in C4H9, how many isomers are possible? 4 structural isomers are possible. Student, this is not last class. These online classes may be continue up to 25th. Now you will get notification on Telegram group. You can join Telegram group. Link is in description. So there we will get all the informations. Maybe it will be extended up to 25th. We'll do some more basic topics of chemistry in these classes. So any doubt in this aldehyde? How many aldehydes are possible? 4 isomers of aldehyde are possible. Okay. Now next is, if we take ketone. Now in ketone, what is representation? It is C double bond O, and on both sides alkyl group will be present. We cannot write hydrogen. On both sides what we have to write? Alkyl group. So you said this is last class. But now there is new notification. Na? We also came to know that we have to continue this up to 25th. So you'll get more basic topics. It will be very good for you. So when you come for offline classes, you will feel very comfortable in classes because all basics you have covered here. Offline also started. Maybe if offline you get notification from I think 18th or something. But student who will come late here. So for those students, it will not be disadvantage. Now RC double bond OR. Now first focus on this question, then I will tell you all about this. Now first start with simple alkyl group CH3. C double bond O. Total 5 carbon atoms are present. 2 we have written. Remaining how many carbon atom? 3. So C3H7. And in C3H7, how many isomers are possible? All we know 2 isomers. One is normal propyl and one is isopropyl. And next, if we take on this side C2H5, C double bond O, then here also what is present? C2H5. And in C2H5 only one structure is possible. There is no other isomer for C2H5. But again, if you write here C3H7, CH3, then this will be identical. Because here both alkyl groups are bonded with C double bond O. So there is no different case. So how many ketones are possible? Only 3 ketones are possible. How many aldehydes are possible? 4 aldehydes are possible. So if I write total number of isomers, so what is answer? 4 plus 3. How much it is? 7. Okay. Any doubt in this? Just like for this aldehyde and ketone, how many total number of isomers are possible? 7 isomers. Okay. So what we have covered? 2 functional groups. Now carboxylic acid ester one case, one is aldehyde and ketone. Now next part will be amide. Now amide is slightly tricky. Similar like amines. In amines what we find? First primary, secondary, tertiary. Similar we have to find in amide, primary, secondary and tertiary. So let us start with amide. Find total number of structural isomers of amide having molecular formula C4H9NO. But we have taken C4H9NO. So we have to find how many total amides are possible. Now before solving this question, first I will tell about representation of amides, primary, secondary, tertiary. One question I will solve for you. Next you have to solve. So let us start with, wait, just I will to check we have written this correct or not. C3H7, C double bond O, NH2, 4, 7, 2, 9, NO, correct. So let us start with primary amide. Now for primary amide, what is fixed part? C double bond O, NH2. This part is fixed for amide. So on this side, what is present? Alkyl group is present. Either R or it can be hydrogen also. But for primary amide, if there are more than one carbon atom, then always here what is present? R is present. So what is representation? R C double bond O NH2. with nitrogen only one carbon is directly attached to our hydrogen atoms. Clear? Next, if we write about secondary amide, in secondary amide, how many carbon are directly attached to nitrogen? 2 carbon atom. So what we write? C double bond O, NH, here R, and here R, or it can be hydrogen. So this is very important part you have to remember. In secondary amide with C double bond O, smallest part can be hydrogen or it can be any other alkyl group. Clear? Third is tertiary amide. In tertiary amide, how many alkyl groups are attached with nitrogen? 2, and 1 carbon is already present. And here it can be R or it can be H. So if you are having notebook and pen, write down these representation, because on the basis of this representation only we can find total number of amide, primary, secondary as well as tertiary. So primary RCO NH2. In secondary, this part is important, you have to focus on this, that here R or H is possible. Same here, R or H. But on this side you cannot change. Here already what is present? Alkyl. Because if you write hydrogen, secondary will be converted to primary. And here also what is present? Both alkyl group. We cannot change this. So if I write for C4H9NO, first I will write primary amide. So in primary amide, what I have to fix? C double bond O NH2. Formula is C4H9NO. So 1 carbon is already involved. How many carbon are left? 3 carbon. So C3, and how many hydrogen are left? 7. Now for C3H7, how many isomers are possible? Only 2 structural isomers are possible. One is normal propyl and one is isopropyl. So if thus, how many primary amide are possible? Only 2 primary amides are possible. Now let us move to secondary amide. So for secondary amide, let us first start with some basic, what we can start, C double bond O, NH, here always R is present. Here it can be R or it can be hydrogen also. So let us start with first hydrogen. R or it can be hydrogen. So I am writing starting with simplest hydrogen. C double bond O, NH. Total 4 carbon atom, 1 I have written. So remaining how many carbon atoms are present? 3. And for C3H7, how many isomers are possible? 2 structural isomers are possible. Now next case is, in place of hydrogen, what I can write? CH3, NH. Now 2 carbon atoms are covered, total 4 carbon atoms. So what is left? C2H5. Now how many isomer are possible? Only 1, because in case of C2H5 there is no structural isomer. Next I will write here C2H5, C double bond O, NH. So 2 plus 1, 3. So only 1 methyl is left. So how many isomers here? Only 1 isomer, because in C2H5 no other structure is possible. Now why these two are different? If you check, here ethyl is attached with nitrogen, here ethyl is attached with C double bond O. So that's why these two compounds are different. So in C2H5 there is no isomerism possible, so only 1 structure is present. Now if I move to tertiary amide, now in tertiary amide, what is representation? C double bond O. On both sides I have to write alkyl with nitrogen. Here we can write hydrogen also. So let us start with simpler hydrogen. Nitrogen, now 2 valency vacant. Now if you check, total 4 carbon atoms are present. 1 we have already written. So how many are present? Only 3 carbon left. So for 3 carbon atom, one is CH3 and one is C2H5. Only this case is possible, because if you change here C2H5 CH3, that will be identical. So how many possible structure? Only 1. Next is, if I write here CH3, C double bond O, N. Here what is present? CH3. Here also what is left? CH3. Because total 4 carbon atom, 1, 2, 3, 4. So how many structure? 1 structure is possible. Now we cannot write here C2H5, because if you write C2H5, So total 3 carbon atoms are present. So only one is left. But how many alkyl we need? 2 alkyl group. So how many secondary amide or tertiary amide are possible? Secondary at count 1, 2, 3, 4. How many tertiary? 2. So if they ask how many total number of isomers are possible, so 2 primary, 4 secondary and 2 tertiary. So what is answer for this total? 8 structural isomers are possible. Check this question. Now this is slightly typical part because we have to find primary, secondary, tertiary. So all possible cases we have to think. Here. Now why this is difficult? Because you have to think about here hydrogen is possible. Here how many combination of 2 alkyl groups are possible. That's why this question is slightly difficult. But if I give another question, if you solve very slowly, first you write all primary, then you write all secondary, then you write all tertiary, then it will be not difficult for you. So I will write a question, you will attempt that question. Find total number of structural isomers of amide having molecular formula C5H11NO. Now this is question for you. Now you have to solve this question and then how to send answer. First you have to tell about primary amide, then you have to tell secondary amide, then you have to tell tertiary amide. So take your time, do not try to do very fast or just type on Google for answer. So first write down for primary amide, then you have to tell secondary, after that tertiary. You have 1 minute or 2 minutes, take your time, then we'll discuss this question. Students are telling primary answer 4. Primary is correct. Now move to secondary and tertiary. Everyone is telling primary correct 4. Very good. Then you have to tell about how many secondary amides are possible. In secondary, you have to apply your mind. Primary, every student can find out. All are telling answer for primary only, 4. You have to tell answer for secondary amide now. Ratna, very good. Very good. Secondary amide are 9. So how many secondary amide are possible? 9, not 6. Ratna, you are correct. 9 are secondary amide. Very good. Now move to third part, tertiary amide. Now students are telling secondary amide 9, correct. Now move to tertiary. How many tertiary are possible? Tertiary, somebody is saying Venugopal 5. Tertiary 5. Very good, Krishna. Tertiary is 5. Good. Very good. Tertiary amide, how many are possible? 5. Now, if you total, how many isomers you will get? Total 18 isomers you will get. Total, how many isomers you will get? 18. All are telling now tertiary 5. Very good. So let us start with first primary amide. We'll discuss all that, how many primary, secondary, tertiary, how to find out. So if we start with primary amide, what we have to fix in primary amide? C double bond O NH2. Now total 5 carbon atom, 1 already written, so how many are left? 4. C4H9. And in C4H9, how many isomers are possible? 4 structural isomers are possible. So if they ask easiest, how many primary are possible, then answer is 4. Let us move to next, secondary amide. Now in case of secondary amide, we have to start from hydrogen. H, C double bond O. Next NH. Now one carbon is present, total 5 carbon atoms, so how many are left? 4. C4H9. And in C4H9, how many isomers are possible? 4 structural isomers are possible. Next, if we move to, in place of hydrogen, if we write CH3, C double bond O NH. Now 2 carbon atom we have written, so how many are left? C3H7. And in C3H7, how many isomers are possible? 2 isomers are possible. Next case, if we take, here what we write? C2H5, C double bond O, NH, C2H5, because total 5 carbon atoms are present, 2, 4 and 5. So in C2H5 only one structure is possible. So it is one structure. Next, if we move, in place of C2H5, if I write C3H7, C double bond O, NH. Now 3 plus 1, 4. So 1 carbon atom is left, CH3. And in C3H7, how many isomers are possible? 2 structural isomers are possible. Now important point is, these two are different. These are not identical. Here propyl group C3H7 is bonded with NH. And here propyl is bonded with C double bond O. So that's why these two are different. So if we count, how many secondary amide are possible? So 4 plus 2, 6, 7, 7 plus 2, 9. So 9 are secondary amide and 4 are primary amide. Now let us move to tertiary amide. So if I have to write tertiary amide, I have to start with hydrogen. C double bond O N. Now 2 alkyl I have to write. 1 carbon is present here, so simplest alkyl is CH3. Now 1 plus 1, 2 carbon we have fixed, and total how many carbon we have? 5. So remaining are 3 carbon atoms, C3H7. And in C3H7, how many isomers are possible? 2 isomers are possible. Next case, in similar, while writing here hydrogen, C double bond O, can I write here C2H5, C2H5? Because total 5 carbon atoms are present. It can be 1 alkyl and 1 propyl, methyl propyl, and both we can write ethyl ethyl also. So how many structure are possible for this? 1 structure is possible. Next, if I take case, in place of hydrogen, now what I write? CH3. So C double bond O N. Now 2 valency are vacant. So total how many carbon atoms are present? 5 carbon atom. 2 we have written, remaining are 3. So remaining 3 means CH3 and 1 is C2H5, where no isomerism is possible. So this is only one structure. Next case I can take, in place of CH3, what I can write? C2H5, C double bond O N. Here what is left now? CH3 and CH3. Total ka 5 carbon atom. So 2, 3, 4, 5. How many are possible structure? 1 possible structure. So if you count total 2, 3, 4, 5, how many tertiary amide are possible? 5 tertiary amide are possible. So total answer for number of isomers are 4 plus 9, 9 plus 5. Total are 18. So if I write answer for total, so total answer will be 4 plus 9 plus 5. How much it is? 18. Any doubt in this? Now check this part. Now this is slightly difficult because total 18 isomers are possible. Now, if you do not know isomers of alkyl and this representation, you will never get answer this 18 isomers. It is only possible if you listen carefully in class, then you remember all the isomers of alkyl group, and next is representation for all the amide, primary, secondary and tertiary. Clear? So this is all about functional groups. Clear? Now let us move to next part. Have you heard about benzene? So we'll write about some benzenoid isomers. So let us write with first question. Find number of benzenoid isomers having molecular formula C4H6Cl2. Or in place of C4H6Cl2, they can write this as disubstituted benzene. But they will call it as disubstituted benzene. Because in benzene, two substituents are present. Which substituents are present? Chlorine. Now try to tell answer that how many isomers are possible for this structural isomer. C4H6Cl2. Take your time, 1 minute, and tell what is answer. And it is easier question. Benzene is fixed because he said that number of benzenoid isomer. Benzene is fixed there. Fix 14. 14, how it is possible? Only benzenoid isomer. On benzene, we have to write two substituents. When we are studying position isomers, we have written this. If you remember, ortho, meta, para, we have written. Remember, ortho, meta, para, we have written. 1, 2 is called as ortho. So benzene is fixed. Na C6. Acha, by mistake I have written 4. I have to write carbon 6 and hydrogen 4. So it is interchange. Na? Acha, that's why you are getting confused. This C6H4, not C4H6. So first, what is possible? 1, 2 at first position, second position. So 1, 2, do you remember? This is also called as ortho derivative. So that is by mistake I have written that. Na? Now next is if we take 1, 3 derivative. 1 chlorine is present at first position, 1 chlorine is present at third position. So 1, 2, 3. And if you remember, we have written 1, 3 is called as which derivative? Meta derivative. So this is meta derivative. And last possible case is 1, 4. 1, 2, 3, 4. It is 1, 4. 1, 4 is para derivative. Now students, wait for 2-3 minutes. We'll complete this topic. Then I will tell you all your queries. When classes will start. What about online, offline? So wait for 2-3 minutes. We'll complete this topic. Then I will tell you all things. So how many isomers are possible for this? Only 3 structural isomers are possible. No, it is not necessary they will write formula. They can write disubstituted benzene. Means 2 substituents are present at benzene. So what is possible? 1, 2, 1, 3, 1, 4. Okay. Now if we take tri-substituted and 3 chlorine are present. If I write next question. Find total number of structural isomers, and in bracket what is written? Benzenoid, means one benzene is fixed. for molecular formula C6H3Cl3. Clear? Apply your mind. C6H3Cl3. So 3 substituents are present here. Now find out how many isomers are possible. It is tri-substituted benzene. So only have to think about what are possible positions for substituent according to IUPAC numbering. Some students are saying 4, some are saying 3. So answer is 3 isomer, not 4. We are talking only about structural isomers. So how many possible cases are there? If benzene is given, first is 1, 2, 3. Means they are present at which position? First, second and third position. Clear? Next possible case is 1, 2, 4. So 1, 2, 3, 4. 1, 2 and 4. Now substituents are same, so there is no alphabetical order or anything. Simple lowest set of locant rule we have to apply. So what is position? 1, 2, 4. And one possible case is 1, 3, 5. 1, 2, 3, 4, 5. So next is 1, 3, 5. Now if you draw any other structure, you will get only these numberings. Either substituent at 1, 2, 3, either substituent at 1, 2, 4 or substituent at 1, 3, 5. Only these 3 are possible. So it means how many structural isomers are possible? 3 structural isomers are possible. Clear? Any doubt in this? Students, if you have drawn any other structure for this, you will get only these numbering. So only possible cases 1, 2, 3, 1, 2, 4 and 1, 3, 5. Very good. So basics of structural isomers we have completed. Clear? Now many students are asking doubts about offline classes. So student, hopefully I think offline classes will start from 18th. If you have not joined Telegram group, join that group. You will get all notifications for that. But with offline classes, we are continuing this online basic classes also. So that will be continued up to 25th. So to continue these classes, only reason is students who will join us late here in offline classes. So they will get some topics of basics here. So what we have discussed, basics of structural isomers we have completed. Now from next class, from last many days we have studying organic chemistry. First Amit sir told about you basics of organic chemistry, then nomenclature, then I came nomenclature and structural isomers. So what we start in next class, we'll move to physical chemistry part. In physical chemistry, we'll start from oxidation, reduction, so basics of that. Okay. So students, thank you for today. We'll meet in next class.
Hello, good morning students. Okay, let us start our session with some questions, few questions. What are the points of crossing over called as? Points of crossing over. Points. They look like X-shaped structures. X-shaped structures or points of crossing over. What are they called as? The answer is paketene and diplotene. Chiasmata are formed here. They are seen clearly in diplotene. They are actually formed in, not zygotene. Chiasmata means points of crossing over. You know, crossing over occurs in paketene. So they are formed in paketene, but they are seen in diplotene. Zygotene bivalents are formed. Zygotene bivalents are formed. Chalo, let us proceed to the today's topic, that is meiosis-II. I hope all of you understood. In the meiosis-I, okay, what happened was a diploid parent cell. Let us take our cell only, 46 chromosomes containing a human cell, or 23 pairs of homologous containing human cell undergoes meiosis-I, and at the end of the meiosis-I, two daughter cells are formed after the cytokinesis 1. The two daughter cells are called dyad of cells. The two daughter cells are called dyad. These dyads of cells which are formed at the end of the meiosis-I will be haploid, because one diploid cell is undergoing the meiosis-I and it will form the dyad of cells, each cell with haploid set of chromosomes. That means n is equal to 23. So now these two cells, the two daughter cells which are formed here, dyad means two, dyad, two daughter cells are formed. These two daughter cells which are haploid, they will enter into meiosis by first passing through the interkinesis. In interkinesis, there is no DNA replication, just the cell will be taking rest for a while and it will be synthesizing some of the proteins which are necessary for the meiosis-II. So there is no DNA replication anywhere in the interkinesis. Just it is like some period of rest in between meiosis-I and meiosis-II. So after the interkinesis, the cell will be entering into the meiosis-II, which will start with karyokinesis 2. In karyokinesis 2 also we have prophase 2, metaphase 2, anaphase 2, telophase 2. So it will be proceeding like normal mitosis. So meiosis-II is actually equal to mitosis. That is why meiosis-II is called as equational division. So in the meiosis-II, first prophase 2. In the prophase 2, what happens actually, the chromatin is condensing into chromosomes. But at the end of the telophase 1, the chromosomes will not be extended like the interphase. Imagine interphase chromosomes are this much, chromatin is this much. But at the end of the telophase 1, they will reach only this much extended form, because they have to continue the meiosis-II into the prophase 2. So they cannot extend fully to the interphase chromosome structure or chromatin structure. So they will be little bit extended, not like the interphase chromatin. So that type of chromatin containing two daughter cells will be entering into the meiosis-II, particularly prophase 2. So they are not fully extended, they are of medium size, the chromatin. So now the further condensation will be beginning in that prophase 2 for the chromatin, and they will become the chromosomes. And here each chromosome will be having two chromatids, because it is like normal mitosis. And that condensation will be proceeding, and you can see here in prophase 2 we have taken n is equal to 2 here, but you can imagine n is equal to 23. If you take 23 like this, it will happen. First let us see for the two chromosomes, because you can see blue one got the pink one and pink one got the blue one. These two daughter cells are having, each one is having haploid daughter cell, each one is having two chromosomes. So both of them are entering into the meiosis-II, karyokinesis 2. So in prophase 2, they will be reaching, they will be condensing little bit, okay. Already they are this much, so they will condense further and to become the exact chromosomes. And the events of prophase of mitosis will happen here, like nucleolus, nuclear envelope, Golgi, ER, all these disappear. If it is an animal cell, even centrioles would have been migrated towards the opposite poles, and they will form the asters, and spindle fibers are also formed here. That is what going to happen in the prophase 2. So all the events of prophase of mitosis will be happening here. The first event, condensation of chromatin to chromosomes. Then centrioles will be migrating towards the opposite poles, and they form the asters. They will give out the microtubules, they will form the asters, and they will be forming the spindle fibers from them. Then the nucleolus, this is the nuclear membrane, Golgi, ER, all of them will disappear, so that these chromosomes will be released into the cytoplasm. Once the chromosomes are released into the cytoplasm, in the metaphase 2, the spindle fibers will come from both the sides, and each chromosome will be connected to the two spindle fibers from opposite poles, like the mitosis. But in case of meiosis-I, if the two chromosomes are like this, okay, these are connected to both the poles by only one spindle fiber. So, only one spindle fiber will be connecting to the chromosome, or you can think that each chromosome is connected to only one pole by spindle fibers 1 or 2. Generally 2 also can be there, but one pole only will be giving the connection to one chromosome. This is in meiosis 1. But here in meiosis 2, in metaphase 2, what happens is each chromosome is connected to both the poles. To both the sides, opposite poles, they are connected. That is the step is equal to the mitosis. So, metaphase 2 is similar to metaphase of mitosis. Okay. So, after the attachment, these spindle fibers will undergo the contraction and relaxation, and they will bring all these chromosomes to the center of this cell. So, once they reach the center of this cell, like this, metaphase plate is formed like this. Here in this cell one metaphase plate, and in this cell also one more metaphase plate. Two metaphase plates are formed because both of them, in both the cells it will be happening. And that metaphase plate will be having all the chromosomes aligned on the equator. And each chromosome is attached to both the poles by two spindle fibers. It is attached, one chromosome attached to both the poles by two spindle fibers. This kind of alignment is important for the proper distribution of chromosomes to the daughter cells. That's it about the metaphase 2. Then in anaphase 2, what happens is, exactly they are placed in the center, no equator. Now the centromere splits. The centromere, which is holding the two chromatids of a chromosome, will split, and that sister chromatids will be separated. Sister chromatids are nothing but the two chromatids which belong to one chromosome. They get separated, and now these are called as daughter chromosomes. Daughter chromosomes. They are formed in the anaphase 2. But in anaphase 1, what happened is just a segregation of homologous chromosomes happened. That means this chromosome is moving to one side, this chromosome is moving to another side. But in anaphase 2, the centromere splits and it results in the formation of two daughter cells. Okay, in the future. So each daughter cell needs one chromosome out of this. So it will form the two daughter chromosomes in the anaphase 2. Because this one will enter into this daughter cell and this one will be entering into this daughter cell. So one chromosome only, it will split into two daughter chromosomes. After that, the cell will be entering into the telophase 2. And all these daughter chromosomes will be migrating towards the opposite poles. Wherever they are formed, this will be going to this end and this will be going to the opposite end. After migration, all of them will be decondensed into chromatin in telophase 2. After migrating towards opposite poles, they will decondense into chromatin. And after that, the nucleolus, nuclear envelope will be formed. So once the nuclear envelope is formed, in the telophase 2, you can see two daughter nuclei are formed. In the cytoplasm, the changes will be Golgi complex will be formed and ER will be formed. Then the cell will enter into cytokinesis 2. Generally, there is a notion that once the meiosis 1, karyokinesis 1 is completed, the cell will not enter into the cytokinesis 1. It's like karyokinesis 1, karyokinesis 2 and cytokinesis. That will happen generally. If not, karyokinesis 1, cytokinesis 1, karyokinesis 2, cytokinesis 2 will happen. So here they have given like 4 daughter cells are formed. This cell will be dividing into 2 daughter cells and this cell will be dividing into 2 daughter cells. Okay, by only one cytokinesis they have taken. But this is also correct. In some of the cells this will happen. In some of the cells karyokinesis 1, cytokinesis 1, karyokinesis 2, cytokinesis 2 will happen. Either way it is correct. But in NCERT they have given karyokinesis 1, cytokinesis 1, then karyokinesis 2 and cytokinesis 2 is given. So by the end of the meiosis 2, we are going to get the 4 daughter cells which are all are haploid. 4 daughter cells are obtained. Each of them are N, and we have started with N is equal to 2 chromosomes, and if you see clearly in each daughter cell also we have 2 chromosomes. The only difference is here the chromosome is having 2 chromatids, here the chromosome is having only 1 chromatid each. So the chromosome number remains same. Here also n is equal to 2 and here also n is equal to 2. As chromosome number is equal, this meiosis-II is called as equational division. But this nickname is given to already mitosis, so it is equal to mitosis. It is similar to mitosis. Okay. So meiosis-I is called as reductional division because if you take the 2 chromosomes, each cell will be getting only 1 chromosome. If you take 4 chromosomes, each cell will be getting 2. If you take 46 chromosomes, each cell will be getting 23 each. So exactly reduction will happen in the meiosis-I, and in meiosis-II no reduction will happen, just equal division, equational division. I hope all of you understood. Mitosis, yeah, it is, mitosis is nothing but formation of 2 daughter cells, okay, which are identical in all aspects. That's what going to happen in the meiosis-II. So meiosis-II is equal to mitosis. Yes, understood? What is the number of chromatids in prophase II? Per chromosome, you can see here 2 chromatids are there, the full chromosome, 2 chromatids. Formation of 2 daughter cells only. Yes, mitosis, formation of 2 daughter cells only, but meiosis, it will form 4 daughter cells. By mitosis, whatever the daughter cells are formed, parent cell, how many chromosomes are there? That many chromosomes are present in the daughter cells. Unlike meiosis. In meiosis, what is happening? If you take the 46 chromosomes, human cell, then the daughter cells are getting only 23. This is usually happening in the formation of gametes. In the formation of gametes. So gametes are generally haploid and they are formed by meiosis. In the remaining all somatic cells, mitosis will happen. Is kinetochore are there in meiosis-II? Yeah, everywhere when I say chromosome, centromere, on both the sides, kinetochores will be there during the cell division. They will come and attach on both the side of the centromeres. They are nothing but disc-shaped structures. They are made up of proteins and they are providing the site for attachment of spindle fibers. Attachment of spindle fibers. Yeah, any further doubts, ma? Any further doubts, students? Then if 2 chromatids split, the daughter cells will have only 1 chromatid. Yes, in the chromosome, only 1 chromatid will be there in the daughter cells. Each chromosome will be having here only 1 chromatid. That's what you can see here, 2 chromatids are there, here only 1 chromatid each. Pink one let us take, okay, this will split exactly in the middle. This will split in the middle and only half will be given to the one daughter cell and another half will be given to next daughter cell. So each chromosome will be splitting exactly in the middle and only 1 chromatid will be given to the daughter cells in the meiosis. No doubt. Okay, very good. So shall I show you one more picture and then we will go with the significance? Okay, you can go through this one more time. Okay, all of you see here, meiosis-II. In the prophase-II, the chromosomes will not reach up to that stage of extension. They will not be fully extended, okay, in the telophase 1. So they will be having medium sized chromosomes and they will be entering into the prophase-II and they will be starting undergoing the condensation process. You can see centrioles are migrating towards opposite poles. Nuclear envelope is broken down and these are migrating towards opposite poles. Nucleolus, nuclear envelope, Golgi, ER disappears. Condensation of chromatin, 2 chromosomes will occur. In meiosis-II, chromosomes line up in the center to form the metaphase plate or equatorial plate. As we have two cells, dyad of cells, two metaphase plates are formed here. Then in anaphase-II, sister chromatids separate due to the splitting of the centromere. Here exactly the centromere will split like this. And now blue one got two sister chromatids got separated. Here also orange one got separated, red one got separated. This is called splitting of centromeres, and sister chromatids are separating. That's what going to happen in the anaphase-II. In telophase-II, the chromosomes would have already migrated towards opposite poles. They will become the chromatin. They will be extending by the decondensation process and they'll become the chromatin. Nucleolus and nuclear envelope reappears there, so that the nucleus will be formed here in the telophase-II. And after that you will be having the Golgi, ER, they will be reformed. After this, cytokinesis 2 will happen. Cytoplasm will divide. If it is a plant cell, it will divide by cell plate method. If it is an animal cell, it divides by furrow method. Already I have told you, cell plate formation. Is it centripetal or centrifugal? It will be going in the centrifugal manner, and furrow will be formed from side to center, towards center. So centripetal growth. That will divide the one cell, this cell into two daughter cells, this cell into two daughter cells. So we are starting the meiosis-II with two cells, dyad of cells, and each cell is forming two daughter cells, which are all haploid. 4 haploid cells are formed, each having only one chromosome of homologous chromosomes pair. Out of a bivalent, only one chromosome is coming to this each cell. So now the cells which are formed, always they will be haploid. And these you can call them as gametes generally, because it occurs in the reproductive cells. These may be gametes or these may be spores, which are generally haploid. Okay, that is about the meiosis-II. Now let us see together what is going to happen in meiosis totally. In meiosis-I, interphase nucleus is there. Interphase nucleus is always a big nucleus, because so many changes will be happening, like G1, S and G2. You know, in S phase DNA replication occurs, G1 and G2 RNA protein synthesis occurs. So because of that, interphase nucleus will be bigger in size and the cell will be taking rest, we will say, but it is metabolically active. And this type of cell, when it enters into the prophase 1, in prophase 1 you have leptotene, zygotene, pachytene, diplotene, diakinesis. There the synapses, process of pairing occurs, nothing but bivalent formation, and then crossing over, exchange of genetic material will happen. Then centrioles will be moving towards the opposite poles, and then the metaphase 1, they are aligned on the center to form the equatorial plate. Now after this, in anaphase 1, the separation of chromosomes occurs. Sorry, sister chromatids are present together. This chromosome will be moving towards this side and this chromosome will be moving towards this side in anaphase 1. It is called, important event, segregation of homologous chromosomes. This occurs in anaphase 1. So all of you have to remember this anaphase 1, because it is important for the segregation of Mendelian factors. You know, genetics, it is linked, anaphase 1, okay. Where the genes or alleles will get separated means anaphase 1. And that process is called segregation of homologous chromosomes of a bivalent. Then in telophase 1, here we have started the cell cycle here with the 4 chromosomes. So in each daughter cell, all of you can see clearly 2 chromosomes only are there. So 4 are becoming 2. So this is the reductional division. Because here we have taken 2n is equal to 4 chromosomes, and here the n is equal to 2 chromosomes are formed in each daughter cell. By the end of the cytokinesis 1, 2 haploid cells are formed because cytoplasm also divides. Now this each daughter cell will be entering into the meiosis-II. It is almost similar to mitosis, so it is equal to mitosis. The question will come where actual reduction occurs. The actual reduction occurs in anaphase 1. Okay, actual reduction occurs in the anaphase 1 of meiosis-I. So in prophase 1, you know, 2 cells will be entering here, and we have started this with n is equal to 2 chromosomes. And in metaphase 2, all of them will be arranged in the equatorial plate, and then in anaphase 2, sister chromatids will separate because of the splitting of centromere. And later in telophase 2, here all of you can see green one and purple one. I hope you can see 2 clearly. So here each daughter cell will be receiving only 2 chromosomes. You can see this green one and purple one. So n is equal to 2. But how many daughter cells we are getting here? 4 daughter cells are formed. 1, 2, 3, 4. In mitosis we are getting only 2 daughter cells, but in meiosis we are getting 4 daughter cells altogether. Okay, which are definitely different among themselves, and they are different to the parent cell also. In mitosis, I told you these daughter cells will be similar to the parent cell, and they are similar among themselves also. But here all these 4 are different because they exchange their genetic material. So all the 4 are different, and these are different from the parent cell also. Okay, because of the crossing over. So here whatever the daughter cells are formed, they are qualitatively, quantitatively totally different. Quality wise also different, quantity wise also they are different, because we have started with 4, these got only 2. So whether you take quantity or quality, they are different among themselves and with the parent cell also. Okay, and these daughter cells further they cannot continue the division. They can't divide. This is an important feature here. These daughter cells can't divide. But mitosis end products, whatever the daughter cells you are getting by mitosis, they can divide. They can continue the division. Further they can undergo. But this is the end of the cell division for them. Cell cycle is completed for them. These ones cannot further divide. Okay. The last topic of today's class, significance. Significance of meiosis. The first point in the significance is, like, you know, crossing over is happening, exchange of genetic material. So that crossing over will lead to variations. Variations means differences among the individuals. So that variations are the sources for evolution. These variations are due to crossing over and recombination. Crossing over will be causing the variations, and variations are the source of evolution, source for evolution. Second one, this is important for conservation of specific chromosome number. In each generation. So you know that we have 2n is equal to 46 chromosomes. So after the meiosis, actually each cell is getting n is equal to 23 chromosomes. But that gametes will undergo the fertilization and they restore that chromosome number. So without meiosis, there is no conservation of same chromosome number in each generation, generation after generations. So this is important for maintaining the constant chromosome number throughout generations. If meiosis is not happening, every time the chromosome number will be doubled. So it is important before fertilization, so that it will reduce exactly the number of chromosomes to half, and then fertilization of 2 gametes will happen that restores the chromosome number. So this is actually maintaining the specific constant chromosome number from generation to next generation. Throughout the generations, it is maintaining that. Okay, these 2 are the important points about significance. In meiosis, A cell with 40 chromosomes in meiosis, a cell with 40 chromosomes entered. Okay. Then after meiosis-I, how many chromosomes are present in the daughter cells? The answer is, you can choose the options: 20, 30, 10. 20. Very good. Continuation for that only. Continuation. Okay, that means that cell is having 40 chromosomes. Okay, now after anaphase 2, yes, option 2 correct. Yeah, after the same question, this one only. After anaphase 2, in the cell, how many chromosomes are present? In the cell, how many chromosomes are present? The options are same options: 40, 20, 30, 10. After anaphase 2. The question is after anaphase 2. That means a cell with 20 chromosomes is entering into meiosis-II. The answer will be 40. First one. You know, 20 chromosomes are having 2 chromatids, right? That 2 will be dividing in the anaphase 2 due to the splitting of centromere, and in anaphase 2 we have 40 chromosomes. Because each daughter cell should get 20 each, no? These 20 chromosomes are having this type of structure. But after anaphase 2, this type of chromosome will split up into each chromosome with 1 chromatid only. So, 1 chromosome is becoming 2. So, 20 chromosomes will become 40 chromosomes. I hope all of you understood. The answer is 40. In meiosis-I, 20 chromosomes are there. Meiosis-I, after the end of the meiosis-I, 20 chromosomes are there. Now, after telophase 1, options are same. The question is after telophase 2. My question is now after telophase 2, how many will be there? Same options. At the end of the telophase 2, each daughter cell, out of this 40, each daughter cell will be getting 10 each. Okay? It will be getting 10 each. Because this 20 got doubled as 40. So, they will be given to each daughter cell with 10 each. 4 daughter cells are formed. So, each daughter cell will be getting 10. A cell with 40 chromosomes enters into meiosis. Each chromosome will be having a structure like this. Okay. After meiosis-I, this 2n is equal to 40. After meiosis-I, it forms the 2 daughter cells where each one will be n. N is equal to 20. Now, each one of them will be dividing and it will be n only, 20 only will be there. Oh, I'm sorry, I have given you 10. No? I have taken the 20 from meiosis. Okay, if you take 20 from meiosis, it will be 10. I have continued the question, no? So I'm sorry. 40 if you take, it will be 20, but I have continued with 20. I have taken in the question, meiosis will be having 20 chromosomes. Then it will become, by the end of this one, it will become the 40 chromosomes and each cell will be getting the 10. So 40 is reduced into 20. If you take 20 here, 20 will become 10 each by meiosis-I, and 10 will become same 10 here. But the difference is, here both the areas, the chromosome will be having 2 chromatids. I have considered this one for the question, so I gave the answer as 10. Maybe you thought that continuation, this is the one. Here chromosome will be having 1 chromatid each. This is the difference. Here pair will be there like this in the parent cell. This chromosome will be given to this and this chromosome will be given to this. So 10, 10 after meiosis-I. After meiosis-II, 10, 10 only will be there, but chromosome structure will be having only 1 chromatid each. Okay. Did you understand here? I can see, okay, all of you see here, this chromosome will be having here pairs. Like this they will be there. Here, each cell will be getting only 1. Here, each cell will be getting chromosome like this. That is the difference. Understood? So, whatever the chromosome, if they give chromosome number, starting, it will be reduced to half in meiosis-I and meiosis-II. At least do you understand now? Do you? Yes, haploid condition, yes. That haploid condition will be beginning with the meiosis-I only, at the end of the meiosis-I only, and it will be continued to meiosis-II. Chromosome structure is important, whether it is having 2 chromatids, whether it is having 1 chromatid. Yes. That's all. Okay, ma. You can read the cell cycle and you can complete that chapter, and whenever the classes starts, you can come and further you will be learning here. I hope all of you understood. Next time if class is there, I will be asking questions based on this. Okay. Thank you. See you. Bye.
So last class we discussed about brain. If your brain or nervous system wants to communicate with other cells, tissues, organs, it depends on neurons, right? Neuron is like a wired phone connection. You know landline phones, right? So if you have to call on another person at another location, you need to dial a number. So there's a wired connection which will convey a message from one location to other location. Yes, but there are some cells, organs and tissues which may not be controlled by nervous system directly. If nervous system cannot have access of these cells, tissues and organs, who would control them? No issues. There was a friend of nervous system who controls those distant cells, tissues and organs. The friend of nervous system which will give wireless communication like Wi-Fi, Bluetooth with the help of hormones. That was endocrine system. Guys, you can imagine nervous system like wired phone connection, whereas endocrine nervous system is like wired phone connection, whereas endocrine system is like wireless phone connection or wireless axis. Okay, so not only with the help of wires, wirelessly also there might be communication possible between cells, tissues and organs. Done. Now, as all of us know that entire body, every organ, every tissue was actually covered and protected by epithelium, right? Sometimes it might be squamous, it might be cuboidal, it might be columnar. It's up to you. They may have cilia, may not have cilia. That's okay. But sometimes epithelium, imagine an epithelium was like this. Linear array of cells. What happens for sometimes, all of a sudden this epithelium layer, it invaginates, it drops down like this. Okay, it gets invaginated, and after invagination these cells of epithelium, they try to secrete some chemicals all of a sudden. So when this invaginated epithelium is secreting a chemical, such kind of epithelium is actually referred as glandular epithelium. What do you call this? Glandular epithelium. During evolution, what happened was, sometimes epithelium got invaginated and remained as such. Sometimes epithelium got invaginated like this. Guys, in some conditions, the invaginated portion was retained, but in some conditions, this part of invagination will be cut off from main layer like this. So the epithelium is left inside your body, where you see an epithelium becoming an organ. Any kind of secretory function, if an organ is performing a secretory function, it could be referred as a gland. This is a gland. Even this part, this can also secrete some chemicals. This is also considered as gland. But this gland is still connected to some layer, and this gland is not connected to any layers. Therefore, every secretion of this gland shall go inside blood supply. So these glandular secretion will mix into the blood. If gland sends all those secretions into blood, because there is no connection now, such kind of gland which is giving chemicals into blood flow, blood supply, was referred as endocrine gland. Whereas here, as there was still some connection, this connection was referred as duct. As there was a connection still retained, all the secretions from this gland, all secretion from this gland will not mix into blood. They will be released into empty spaces of your body. They might be released into buccal cavity, they might be released into stomach, They might be released into intestinal chambers like this. So these glands, they will not secrete their secretions into blood. They will have secretions out of blood flow. Therefore, these glands which do not secrete into the blood, which secrete into some empty cavity, was referred as, what do you call them? Exocrine gland. Now you got nomenclative explanation. Exo, outside blood. Endo, into the blood. Endo means into the blood supply. Exo means outside the blood supply. Therefore, exocrine glands, actually they possess ducts, so we also call them as ductus glands. Whereas endocrine glands, as they do not possess any ducts, we could refer them as ductless glands. Done. Exocrine ductus, endocrine ductless glands. Most of the secretions of exocrine glands, they usually consist of enzymes. Usually, secretions from exocrine glands may possess enzymes, very common. Whereas a fluid coming out of endocrine glands will not have any enzymes. Secretions of endocrine gland will possess hormones. See, exocrine ductus gland secretes enzymes. Endocrine ductus glands, they will be secreting hormones. So exocrine glands are ductus, endocrine are ductless. Exocrine gland secretions possess enzymes, whereas endocrine gland secretions possess hormones. Now a simple question for you. Give me some examples of exocrine glands which have ducts. Ductus. Give me some examples of exocrine glands. Yes, start now. Come on. No pancreas. Not completely accepted. Salivary glands, very good. I'll give you some examples. Okay, salivary glands. Okay, sebaceous glands. Done. Again, salivary and liver, very good, largest gland of your body. Liver is actually largest gland of your body. Among exocrine and endocrine combinedly, largest gland was always liver. One more. Sweat glands, very good, sweat glands. One more. Tears, tear glands, we call them as lacrimal glands. Lacrimal glands, tear glands for tears. And one more, mammary glands for ejection of milk. This is also an example. Even more, come. Even more. Yes, digestive glands, very good. Gastric glands, intestinal glands, Brunner's gland, many examples for that. One more. Lacrimal done. Thyroid gland, no. Pituitary, no. Mucous glands, yes, we can write it. Adrenal, no. One more was there. Try it. This gland was found in your ears, secreting wax, ear wax glands. Yes, Ratnabujji, Kuchpudi, liver does not secrete enzymes, but it has activators for every body process. Yes, it is exocrine, but usually liver secretion do not get an enzyme. Bile has no enzymes. Yes, correct. One more. Wax glands of your ears. That is enough. That is enough. Okay. Now, tell me examples of endocrine glands. Give me examples of endocrine glands. So, the endocrine gland which was found outside of male body and in abdomen, lower abdomen region of females. In males, there are testes. In females, they have ovaries. Apart from acting as primary sex organs, these are also endocrine glands, and combinedly we refer them as gonads. Male gonads are testes, female gonads are ovaries. Just if you go a bit above, adjacent to kidneys, there was a pair of glands. Those glands adjacent to kidney was adrenal glands. Even more, below stomach, there was a gland which was 98% exocrine but 2% endocrine. We call that as mixed gland actually. So that was pancreas, single gland pancreas. Guys, pancreas, singular. There are no two pancreas. There is no term like pancreas. No, pancreas itself is a single gland. There is no term like pancreases. No, just pancreas. Bit above, just adjacent to your heart and lungs, there was a gland which can decide how much you live, your lifespan also. Which gland was that? Which is deciding your immunity? Thymus. Very good. And coming to neck region now. Largest endocrine gland of your body, which looks like butterfly. We often call that as butterfly gland. What was that butterfly shaped gland? Thyroid. And adjacent to this butterfly like gland, there are 4 more attached to it on back side of this. What do you call this? Parathyroid. Right? And most of these glands will be directly or indirectly controlled by a gland inside your brain, that was pituitary. Often pituitary was regarded as master gland because it controls many other glands. And just above pituitary or adjacent to pituitary, there was a gland which decides when you sleep, when you wake up, what you do, your immune system, your menstrual cycles, and this gland sets up biological clock of your body. Time to sleep, time to wake up, time to work, all these things were automatically set by this gland. The gland which is also considered as third eye of human body. The gland which is also deemed as most important gland responsible for maintenance of rhythm of your body, lifespan of your body, that was pineal gland. And last but not least, all these glands, every gland
was actually under the control of brain, but entire brain does not control it. A part of diencephalon controls it. As I told yesterday, diencephalon has three parts: epithalamus, thalamus, hypothalamus. So the part of brain which controls all glands, hypothalamus. Grandmaster, grandfather, absolute leader of all glands. Even pituitary has to listen to hypothalamus. You can see the list, right? Hypothalamus, pineal, pituitary, thyroid, parathyroid, thymus, pancreas, adrenal, testis or ovaries. These are gonads. Yes, Deeptika. Hypothalamus controls pituitary gland and pituitary controls every other glands. Yes, master gland is hypothalamus. Exactly, Nagamani. Good. Yes, exactly. Very correct. So a question for you now. Just look at this list and I have got a question. Shall I ask you? Among these 9-10 glands, which gland is acting as both sex organ and gland also? Which is both a sex organ and gland also? The gland, that organ makes gametes, and that organ can also make hormones. What are those? Gonads. Very correct. Yes. Good. Yes. Next question. Very correct. Ovaries and testes. Ovaries make female gametes and ovaries manufacture progesterone, estrogen. Same time, testes manufacture male gametes, spermatozoa, and they also make male sex hormone testosterone. Very good. Correct. Next question. Which gland is responsible for increase in energy, increase in sweating, increase in your alertness when you are in stressed condition, when you are excited? Guys, when you are stressed, excited, frightened, amazed, shocked, hormone will be there which will be doing all these things for you. This hormone was adrenaline, which is now called as epinephrine. And which gland makes this? The gland responsible for your stress condition. Yes, flight or flight. Adrenal glands. Very correct. Correct. Next question for you. Among these glands, which gland secretes out insulin so that glucose in your digested food will get absorbed into body? Which gland secretes insulin which helps in sending glucose of digested food into body tissues? Very correct. Pancreas. Accepted. Pancreas. Very correct. So pancreas pushes glucose and blood into body parts. Therefore, insulin decreases blood glucose level. That's how it does. Done. Next question. Among these, this gland is very temporary. It will live in your body till 32 maximum, 28, 30, average 26 to 30 years. That's it. After 30 years of your age, at least 30, 32, this gland will automatically die. This is a temporary gland of your body. It dies very soon. What was that? And this gland also decides your immune system lifespan also. Which was that? Thymus. Very good. Thymus is a temporary gland. In all mammals, thymus will be died very soon. Once you cross your adult age, once you reach 25, 26 till 30 years of your age, thymus will slowly decrease in size and finally disappears. An adult body who has crossed 35 years of age will not have thymus. Therefore, they are more prone to diseases. Young individuals like you are not prone to diseases. You are very immune because you have functional thymus. But old people get more diseases because they do not have functional thymus. Correct? Now, done with this? And few more questions. This is a gland which is also part of a brain, part of forebrain. This gland is considered as master of master gland or grand master of your body, and this gland can control hunger, thirst, emotions, temperature also. What was this grand master gland of your body? That was hypothalamus. Very correct, very correct. Very good. And among these again, the gland which is found inside your brain, size of a pea, and this gland is considered as third eye, and this gland determines when you sleep, when you wake up, when you work, when you rest. This gland will inform you when should you go under shade, when should you go into light. This gland has also effects on your immune system. This gland can control menstruation in females. This gland can also control pigmentation of your skin, and this gland does it by making a hormone known as melatonin. Before 4 years, a scientist was awarded Nobel Prize for his research on this gland. This gland sets your biological clock. You don't need physical clocks. You don't need physical clock to maintain certain timings. You need to listen to this gland. That's it. This gland sets up your biological clock. That is third eye. And this gland will be less functional in old people, more functional in young people. But it always lives. It lives completely. This gland was third eye of your body. That was pineal. Yes. I mean same thing. Nagamani, yes, it is maintaining circadian cycles, biological cycles, night day cycles, wake up cycles, correct, sleep cycles. You know that if you all of a sudden, if I take you to some USA country or Canada, Alaska, there is a time gap of almost 14-13 hours, right? If all of a sudden you fly to another country which is far away from our time zone for a few days, you may feel some disturbance in sleep and wake up time, because for now, right now it is daytime for us, but at the same time it was night time for USA people, right? When you change your time zone like that, definitely you will have a very severe headache and very severe adaptive differences in time zones. You may then after going to that country, you may sleep at daytime and wake up at night time, just like Indians. So very slowly you get adapted to the country's time zone. This difference in time zone will cause you some disturbance in your metabolism, and this is caused by whom? Which gland? What do you call this phenomenon? Jet lag. This jet lag will be slowly compensated by pineal gland. Correct? Very good. Now, 3 more questions are left for you. Gland, if your food does not contain enough iodine, this gland swells up, your neck may swell and looks very odd. Swelling of neck was referred as goiter. If your food does not have enough iodine, this gland may not like it. Which gland was this? It is thyroid. Okay, These are glands which we'll be learning in detail once you come here. Guys, among these glands, which is the largest of endocrine glands? Of course, thyroid. It was often referred as butterfly gland because of its shape like this. This gland has two lobes, lobe 1, lobe 2, and these two lobes are connected by a tissue. This tissue is like a bridge between two lobes. This bridge-like tissue is known as isthmus. Okay? Guys, thyroid gland is one of the most vital glands of mammals because thyroid gland can secrete two types of hormones. It secretes thyroxine and one more hormone was thyrocalcitonin. There are two hormones coming out of thyroid gland, thyroxine and thyrocalcitonin. And of this, thyroxine maintains RBC production, thyroxine maintains level of metabolism in a body, thyroxine will manipulate your body systems to behave properly. Thyroxine maintains the metabolism of your carbohydrates, lipids and proteins also. Thyroxine will decide what will be the fate of carbs you eat, what will be the fate of lipids you store, what will be the fate of proteins you want to use. That was the function given to thyroxine. And if you have got low levels of thyroxine, obviously metabolism may get decreased. Your neck may get swollen, leading to simple goiter. If it happens to you, that is goiter. Okay, accepted. But there is even more worse condition. Guys, listen. In case of pregnant females, without an exception, almost all of those pregnant females and mammals, they will have elevated, increased level of hormones in their blood. So pregnant females, when they get their blood checked for hormones, every hormone will be in more concentration. Every hormone will be hyper secreted because they have to support their own body as well as they should support their baby also. So pregnant females are not supposed to go through any hormonal check and get any disease detected. No, that was not absolute thing. Of course, they will have more hormone. That doesn't mean they have some disease. That's normal. So they have all those hormones in more concentration. But by any means, if pregnant females have got less amount of thyroxine in their blood, if thyroxine is very less in their blood, entire thyroxine will be used up by them. They will not send any thyroxine to baby. Inside baby, that fetus, lipid, protein, carb, DNA, RNA metabolism will be disturbed. That fetus will have very poor development of their brains. Myelin sheath is not properly formed. Skin pigmentation is not properly done. Their bones get weakened. Their height will not increase later in stages. They will become mentally retarded also after birth. If pregnant female has hypothyroidism condition, that is very, very dangerous for her baby. What do you call such condition? Baby born to her will have stunted growth, short stature, mentally retarded, congenital defects in their body parts, disturbance in metabolic events. We refer to those conditions as cretinism. That's why we always say pregnant females to eat properly, eat enough amount of diet, take salt. Salt has iodine. If you have enough iodine, you may not go through thyroxine deficiency. That was a precaution to pregnant females, like this. There are many more aspects to be discovered. And my question for you today, for tomorrow. So guys, this was a mystery question till now. This is a mystery question till now. If you can give me answer, that will be very good and excellent from your side. I'll just ask question from this only. Okay. The mysterious question was, among all these glands, which gland it makes both enzymes for helping in digestion and also makes hormones also for regulation of glucose? This gland makes both enzymes and hormones. What was this gland? Question. Yes guys, pancreas. Very correct. Pancreas. Now coming one more mystery question. Very good, very good. You're listening. Very nice. That's good. Very correct, very correct. So most of you are listening. Very correct, very nice. One more question. Among all these, deficiency of iodine first hits which gland? Which gland responds to deficiency of iodine first? If your food does not consist of enough iodine, this gland will be severely affected. If she was pregnant female, her baby will also be severely, irreversibly affected. Cretinism was irreversible. You cannot change it. You can treat goiter, but not cretinism. What was that? Thyroid. Very correct. Done. So there is one hypothalamus, one pineal gland, one pituitary, one thyroid, but there are 4 parathyroid glands. Single thymus, single pancreas, 2 glands and 2 testes, 2 ovaries. Highest number of glands, of course, parathyroid. How many are there? 4. 2 pairs. Attached to thyroid, 1 pair on left lobe, 1 pair on right lobe. Like this. Parathyroids are 4 in number. Adrenal 2 in number. Testis 2, ovaries 2. Remaining every gland was single copy. Done? So we'll be concluding here. We'll meet very soon. Okay? So please stay indoors, stay safe, stay hydrated, eat good food, eat sensibly, drink more water, eat less junk food. I'm not saying do not eat junk food, eat, but less. Okay? We'll meet tomorrow. Thank you.